Hey guys, hi. So we're starting with human anatomy and physiology, and let's start with a very basic, that's tissue. So what's tissue? You already know it's a group of cells, right? Now these cells must have similar, not same, they will have similar structure, and if not same or if not similar function, at least they will have related functions, right? So they will have similar or related functions. So in animals or humans, because we will be largely focusing on humans, we have four different types of tissues, and that would be epithelial, connective, nervous, or muscular. Now, if you want to have a very generic idea of what these do, so epithelial tissues, they generally cover your body, right, on the outer and the interior. So they cover on the out and the in of the body. But as we go on into the detail, you understand that they perform a lot of different functions of protection and what not, right? The next is connective. Connective tissues support your body, so your ligaments, tendons, even your blood is connective tissue. So remember that these are only very generalized descriptions of the same. Nervous tissue control, so your brain, it's largely made up of nervous tissue. It thinks what you're seeing right now, what you're hearing, and makes sense of all of that. Muscular tissue, on the other hand, moves. So right now I'm speaking because the muscles in the mouth are shaping the vibrations that's coming from my lungs and producing a sound, right? So from walking to speaking to eating, a lot of internal and external movement are because of the muscular tissue. Now we are going to start with the epithelial tissue, and we'll try to cover in as much depth as possible. Now, as you already understood, that the large function of the epithelial tissue is that it is going to cover the body on the outside, and it will also line the internal body cavities. Very important. So they because they line internal body cavities, they are not limited only to the function of protection. So general function that you would expect from this is protection, but besides they cover the internal, because they can cover the internal body cavities, they also play a role in absorption. So you eat food, you digest it out, and then they go inside, and the stomach is lined by this epithelial tissue as well. Now excretion, so they might secrete some waste materials, like it happens in your kidneys. And secretion, now please don't get confused on excretion and secretion. Excretion is your pushing out the waste product. Secretion, on the other hand, is you are secreting saliva that is going to digest, you're secreting some tears, you're secreting some hormones or whatever, right? So that is secretion. And last but not the least, it is also going to perform sensory function. Now the moment I say sensory function, you should start thinking about your nasal epithelium, the epithelium that's right there in your nose and performs a singular function, not singular but very important function of sensing the aromas around you, the food smell, or it could be a perfume or whatever, right? Now let's talk about the special characteristics that epithelial has. Now let's talk about some of the features which are very generic to all types of epithelia that we'll be studying about. The first thing is cellularity. That means epithelial tissue is composed largely and mostly, if I should say, only of cells. That means little or no extracellular matrix. That means there would be nothing cementing between the cells. It's just tightly packed cells, and there would be minimum extracellular matrix. Polarity, that means there will be two types of surfaces. So if this is your epithelial tissue, there would be one surface that would be exposed. So this is, let's say, a tube. So if you take a section of a tube, there would be a surface that is exposed on the outside. So that surface you can easily call as the apical surface, and there's a basal surface. Now why we say there is a polarity? Because the distribution of stuff is different. Sometimes you'll find that the nuclei will always be towards the base, right? So that is why we say there's a polarity. They have different properties. The apical surface could be also modified. Now, modified in what aspect? Because we understood in the last slide that they can also perform the function of absorption, right? And modification can be of two types: microvilli. So if this is a cell, the upper plasma membrane would form these minute finger-like projections. Now, you'll be surprised to know that inside your stomach, this specific feature increases the surface area around what, 200 times, or is it 600 times, which is around the area of a tennis court or something inside your intestine. So it's because of this fold. Now, some people might have hard time saying that how is this increasing the area. Now, the cell could have been just this, right? And in that case, what would be the area? Just this much. But now what is happening is these stuff is, they are also going to absorb stuff, right? So that is how the folds increase the area. It's a very simple stuff. Okay, now what else? They could be modified to bear cilia. Now the function of cilia is pretty simple. They are hair-like structures, and these hair-like structures, they can move. Now when they move, they'll move the substances that are around, right? They can work to move a fluid. This happens, example for they will move mucus. Mucus is like a gooey, slimy substance that's lining a lot of cavities inside you. So your windpipe, that has a lot of mucus, which keeps on trapping the bad particles and pathogens, and the cilia keeps on moving, and they're pretty important. For example, they trap things, and when people smoke, they kill the cilia, and the mucus keeps on accumulating, and then they keep on coughing, trying to just push it out. Okay, so that is how it's pretty important. So apical and basal layer. Now when you come back and just go about this, you will also find that beneath the basal layer there is another something that is called the basement membrane. One of the key properties of your epithelial tissue, this basement membrane is acellular. That means there are no cells. It's a simple molecular layer which is largely made up of collagen or and glycoproteins. So it's glycoproteins, if you're confused about that, proteins that have carbohydrates attached to them, those are glycoproteins, right? So it's acellular layer that's is there just beneath the epithelial tissue. Now what's the function of this basement membrane is pretty important. The next property we'll be talking about is avascular. I'll come back to this. So epithelial tissue is avascular. That means no blood vessel, right? No blood vessel enters the tissue. So this is where the basement membrane comes in. So the basement membrane would act as a filter, right? So it will decide which substances are going to diffuse across the basement membrane and be available to the epithelia. Okay, so it's a filter, it's a gatekeeper, right? So the blood vessels would be beneath the basement membrane in the underlying connective tissue, and this basement membrane is going to filter and decide. It also serves as an attachment, right? It attaches this epithelial tissue to the underlying connective tissue. Just underneath the epithelial tissue, you'll have the connective tissue. So attachment point is this. Okay, we already talked about the basement membrane right here. So that's done. Avascular, no blood vessel, very good. Extensive innervations. What does this mean? Innervation means nerves. Now, generally, your epithelia is richly supplied with nerves because they would be able to detect a lot of changes in the environment and report back to brain, in which case it will analyze and give a command. High regeneration capacity. This is something we observe in daily life. So very often or not, you go and bruise yourself, maybe while playing football, or you simply can't walk straight and you fell or something like that. So a lot of time, because of that and because of simply eating, when you eat, you erode the layers of the epithelia that lines your mouth, right? All of that happens. So because of few stem cells, the deepest layers, if there are multiple layers, the deepest layers of epithelia will have stem cells. Now these stem cells will divide and keep on producing a new batch, right? They will keep on producing a new batch of cells which will help you regenerate the lost part. So these are the general features that you would be seeing in each and every type of epithelium that we will come across. Okay, now we are going to talk about the classification of epithelial tissues, guys. One request, I would request you to try to watch the videos in the highest quality settings, because it's already recorded in 180p and it's a lot of scribbling that is going to happen. And so if you're watching in lower quality setting, it would hinder some text or some drawing, so maybe that would be best. Also, make sure that you like, share and subscribe the videos. Okay, thank you. Now, classification. We have to classify the epithelial tissues, and the classification happens on two bases. What are the two bases? First, obviously, is going to be shape, and the second is going to be the number of layers. Now, let's go with the number of layers first. Easy. So, simple epithelium. Simple means there's just one layer, right? So, that's it. One layer of cells. Then you have stratified. Stratified, the word comes from strata, that means more than one layers. So you'll have, now it's slightly time consuming to draw multiple layers, right? But that is stratified. There's a third type, and this is where things get a little bit murkier. Pseudostratified. Now, what does pseudostratified means? So all it means is that there's actually just one layer of cells. So let me first draw basement membrane, and then the layer of cells, it's just one. As you can see, right? There's just one layer of cells, but there's no consistency in the shape. Because there's no consistency in the shape, the nuclei happen to be at different depths. So when it was first observed under the microscope, it gave an impression that it's more than one. But later on it was understood, well, no, that's cheating. It's just one layer of cells, right? So that is why they are called pseudostratified. So this is one type of classification. The second is shape. Now I want to make one thing very clear. There's two ways to look at it. First, you look at it from the top. When you look these cells from the top, they would all appear slightly pentagonal in nature, attached to one another very closely. These are not pentagons, but slightly they would appear like that. But this is the top view. Things get interesting when you have a lateral view. What does lateral view mean? You're seeing it from the side. So when you see it from side, you have the following shapes. So the very first shape, which is very easily identifiable, is squamous, which means plate-like cells. So cells are very flat, plate-like. Now, next category, as you can see, is cuboidal. That means the cells are shaped like square from the side. And the next category is columnar, which means they are shaped like they are more in length than they are in width. So let me just write L is greater than B in mathematical terms. There's one type which is found very at selective places. It's called transitional. Now what does transitional means? That they are usually polyhedral, they would be somewhat like rough squares, so they're usually polyhedral, but when stretched they very easily become flatter, right? They become more flat. This happens in urinary bladder. Now as you can imagine, the task of the urinary bladder is to store urine, and you're out from home to a place where there are no washrooms, you drunk plenty of water, and now your urinary bladder has to take the hit. So what it does is it has to accommodate more and more urine, and one of the ways that it has worked is that these transitional epithelium, they change shapes. Okay, now as you can imagine that we have to club the two classification systems, that is the basis of shape and the basis of layers, and come up with a more diverse system, right? So let us try to do exactly that. So combining the two classification systems, so you can come up with a lot of variety. So first you divide them into simple and stratified, then divide each category according to the shape. So you can have simple squamous, cuboidal, columnar, simple columnar, and pseudostratified columnar. And similarly, if you go to the other end, you can have stratified squamous, stratified cuboidal, stratified columnar, and transitional. Also, stratified squamous could be keratinized or non-keratinized. What does that mean? Don't worry about it. Your skin is keratinized. It has the dead pigment keratin, and there's non-keratinized squamous as well. We'll talk about that. Okay, so our job is to do two important stuff. First, we have to understand the function of each of these types, what exactly they do, right? And second, you have to try to remember the location of each. Now if you remember the function of each of these types, you don't have to remember, you'll understand the function of each of these types, it would be easier to pinpoint the location, because you already know the function, you already know grossly how your body works, where what is happening, so you'd be able to do that. Okay, so let's get started. So let's get started with simple squamous. Okay, so simple squamous, as you already know, the characteristics, it's flat, thin cells. Now what can flat thin cells function for, right? Absorption or exchange, right? Because it's flat and thin, it could be easily transversed. Things can cross them very easily. So you can expect that the location of simple squamous is where alveoli of lungs. What are alveoli? So your lungs has the windpipe, you know, bisects one entering each of the lungs. There it goes and divides and forms finest tubes, and later on each tube gives rise to these sacs. So one sac in here is the alveoli. So this one sac, just a moment, this one sac, this is alveoli. So air reaches here and it has to cross the alveoli into the blood vessels, right? So the alveoli in the lungs has simple squamous. What else? The blood vessels. The finest blood vessels, also not just the finest blood vessels, but even the other blood vessels, the innermost lining will be your simple squamous. That also allows exchange. It will also be found in the serous membrane. Now don't worry if you don't understand quite exactly what serous membrane. It's the covering of the internal organs. I'll go into it detail, but that is also will you find simple squamous because we need exchange happening. The next category is simple cuboidal. Okay, now what function does cuboidal have? Now because they are very uniformly made, they form good structural components, right? They're structural, they can easily form structural components, and that is why their location, they form they are in the wall of small ducts. So you'll find them there. It can perform structures, it can be structures. It can also function in some absorption or secretion roles, even that is possible, right? So you'll easily find them in the small ducts, in your kidney tubules. So kidneys have these tubules, small, extremely fine tubes that you call nephrons. You'll find them there. Now we jump to the next category, that is simple. Now, simple columnar, I am using the red color just to distinguish. Let's use this. I'll draw a line later on. So simple columnar could be of two types. Now they're columnar, that means they're long. They're more long than they're wide. So they generally have the functions of absorption, little secretion as well. Now you would wonder, if they are so good at absorption, I mean, then why do we have simple squamous? I mean, come on, you can't add absorption at everything, right? So hang on for a while, this will make sense. So simple columnar is actually of two types. Now remember microvilli and ciliated, so that this is where they come from. So some of the simple columnar, they have a lot of microvilli, right? And under the microscope they appear as brush, and that is why some books they will give you brush border epithelium. It's nothing but is a simple columnar with the microvilli, and there they would increase the area of absorption, right? Now generally wherever this is present, they'll have goblet cells within them. Within them means interspersed, and these goblet cells, they secrete a specific substance called mucin. Now this mucin, they mix with water when hydrated. That is what forms mucus, right? The slimy coating over everything. Now besides having microvilli, some of them could have cilia, so they would be ciliated simple columnar epithelium. Now you can expect what they do, they will move substances, right? So now that you know the function, you can guess the location very easily. So microvilli, that means you'll find them all over intestine. That is where you need the maximum absorption. Now ciliated one, you'll find it very easily over larger bronchioles, right? So you'll find them over larger bronchioles. You'll also find them in uterine tube, because they have to move the ova that had been secreted by the ovary. So in uterine tubes as well, you'll find them. The next and the last category in the simple is pseudostratified columnar. Now pseudostratified columnar could also be of two types. What are those two types? Those could be ciliated and non-ciliated. Now ciliated is generally found in larger air passages. Now larger air passages mean the trachea, some parts of pharynx, some parts of nose, right? There you'll have pseudostratified columnar. They will also secrete mucus. Why? Because they got to protect that part. Guys, I have one question actually. Please let me know if you want bigger videos or smaller videos, because depending upon the length of the video that you're comfortable watching, I would divide the concepts according to that. So this video is, I think, is going to turn out pretty big, but as per your requirement, I'll change the things that happen in future. Okay. Now, non-ciliated columnar epithelium. What's the function of that? So non-ciliated columnar is sort of a little rare, right? They don't have goblet cells. So no goblet cells, and that means no protective mucus or anything like that. But where are they present? They would be present in male urethra. If you are not aware what's urethra, it's the tube that takes the urine from the urinary bladder out to the body. It might also be found in epididymis. Once again, if you're not aware of this term, don't worry. Epididymis is a short tube that takes the sperm and stores temporarily before passing it down to the vas deferens, which carries it inside the body from the testes. Don't worry, we'll talk about them later on, right? So these were the broad classification schemes of the epithelium. Okay, now we are going to jump to the stratified epithelia. Okay, stratified squamous, stratified cuboidal, columnar, and transitional. This is going to be easy. Now, squamous, you already know, plate-like. Now, stratified means there's multiple layers. Multiple layers means they'll do the job of protection, right? So that's their most important job, that is protection. Now, depending upon how much of abrasion, wear and tear is there, they could be keratinized or non-keratinized. Keratinized, the common idea is the skin, right? So they have stem cells in the deeper layers which keep on producing new cellular layers and layers and layers, and eventually they get keratinized. A protein called keratin gets deposited, and this keratin uh, eventually makes it tough and also dead, right? Then you have non-keratinized stratified squamous epithelium. They'll form the surface of the mucous membranes, right? Where mucous membranes in the mouth, the oral cavity, they'll be there in pharynx. So all such cavities where you can't afford a dead layer. So internal layering, some parts of even pharynx, esophagus. Esophagus is a food pipe, the pipe that conducts the food right from the mouth to the stomach. So all these places you'll find stratified non-keratinized squamous epithelium. Now, stratified cuboidal epithelium. Once again, the basic funda is clear. It has more than one layer, two or more, obviously, and it's cuboidal in look. What will it do? Has similar function. It will do the job of protection and some secretion. That's the generic function, right? Now, where you'll find them? You'll find them in the ducts. Ducts are tubes of glands. So most exocrine glands, there you'll find these guys. Also, exocrine glands like sweat glands, right? So you'll find them there. Okay. Now, stratified columnar epithelium. This is relatively very rare in the body, right? And there's only a few places that you'll find them. So large ducts of glands, so glands which have large ducts, you'll find them there. You'll find them in the conjunctiva of the eye, okay, and a part of male urethra. But I think remembering large ducts is sufficient. Finally, transitional epithelium, which we have already talked about, that the whole function is it allows for adjusted storage in the urinary bladder because it can change the shape. So guys, this is where I think we are hitting a pause on these videos, right? Just to give you a summary of what we did today. So we started by understanding what's a tissue, what are the four types of tissues and their very generic understanding. Epithelial covers, connective supports, nervous connects, uh, and then muscles moves, right? And then we started with epithelial tissues. We talked about the general features, that they have a polarity, that they have little or no ECM, extracellular matrix. They also have a basement membrane. They have too much nerves, and they are avascular, and they have high regenerative capacity. So we talked about those general features. Then we talked about the classification, right? Classification we can talk about classification in terms of number of layers or the shape. Layers may we can have simple or stratified, more than one layer. Shape may we can have squamous, plate like, cuboidal, square like, columnar, which is longer, and transitional. Then we talked about each of the type, and we talked about various functions, locations, and stuff. Okay, so in the next lecture on in the same series, we'll be talking about glands. And if do remember to like and share the video and subscribe to the channel, guys, because that's a huge help. Also, if you're wondering about how we're going to do this, so we'll have different playlists. One would be human anatomy and physiology, another would be cell biology. We'll start with genetics soon, microbiology and other stuff. So we'll start with different playlists, and we'll have videos uploaded regularly so that you can flip back and forth, and I'll try to organize it as much as possible. But until then, hope this helps. Thank you so much for watching, and uh, yeah, thank you so much. Bye bye, take care.